In this module, we will discuss the ligand-gated ion channels, that is the LGICS, which cell uses to communicate with the other cells. Now, group of transmembrane ion channels, proteins, which open to allow ions such as sodium, potassium, calcium, and chloride to pass through the membrane in response to the binding of a chemical messenger, that is a ligand. Chemical messenger is known as a ligand, and such as neurotransmitters, which are used for the communication of the cells through the membranes. Now, mind you that the membranes are there to separate the outer side of the cell and the inner side of the cell, and some of these ions, they actually are inducing the charges, other are increasing the influx of the potassium, which causes hypertension. Similarly, enzymes are there which go into the cell through these pores, and these pores are regulated by the ligand, that is the chemical processes. Now, channel families which are used, there are simple pores like GA, GAP junction, substrate gated channels, which is the nicotinic receptors. These are the chemicals. Now, voltage gated channel, as we discussed, like potassium channels, and different pumps like ATP synthesis, potassium, sodium, and ATPase. These are known as the channel families in the membrane. Now, in this cartoon, the cationic channels, for example, for potassium, sodium, and calcium, and nicotinic, they are just shown that how they act. There is a messenger which goes onto the cell, the binding site is there, and then it is further induced to the cells, and the cell messenger or the other components, they pass through the membrane for the desired effect or the desired signals. Now, as they are ligand controlled, so some biochemical changes that lead to the channel gating or opening or closing. Now, these channels, they are just like just the ways where they are closed or open. And how that is effective, that is the gating involves some type of conformational changes. Remember, during this discussion, we have always been discussing that in the chemical ways, the conformational changes, that is the molecule itself, it goes into the change and produces the desired effect. So in the conformational changes in the proteins, and there are several general purpose models for the gating, that how these channels are opened or closed, the different models they are suggested. Now, conformational changes, they occur in discrete area of the channel, that is in the channel, not throughout the channel, but in the discrete area and leading to its opening. ये एक सिरे पे हो सकती है या दूसरे सिरे पे हो सकता है, and the channel is open. The entire channel it changes conformation. इसकी conformation जो है, इसकी माहियत जो है, वो बदल जाती है. फॉर एग्जाम्पल इलेक्ट्रिकल सिनॉप्सिस, इसमें इलेक्ट्रिकल पोटेंशियल जो है वो चेंज हो जाता है, एंड देयर आर डिफरेंट मॉडल्स लाइक बॉल एंड चेन टाइप, वेयर इन द चैनल देयर इज a ball and chain, that is the ball actually it fits into the channel or it is removed on the channel, and the ions or the other signals they pass through. Neurotransmitters, they are hormones, are the binding causes of the channel to open. Some of the neurotransmitters, they are hormones, and some of them are not, but they are the mainly responsible for the channel to open or close so that the desired effect can be carried out. Now, this is the ball and chain like mechanism for ion channels as we discussed. You can see there is a ball and a chain. When the channel is closed, the blue area is closed and the ball is at one discrete end. When it is opened, the ball is removed and the channel is open. And further is because all these channels, they cannot be opened or closed permanently. So we have to inactivate that. Cell has to inactivate that. And when it is inactivated, the ball, it fits into the channel. And remember, again, they are at the discrete end of the channel. And this is the ball and chain model. Now, neurotransmitters, which are hormones, they are binding to the receptors. Now, there ought to be a receptor which should receive the signal. And what it is, the signal causes a second messenger to activate a protein kinase that phosphorylates a channel and thus opens it. So it is phosphorylation and then channel, it opens it. Now, changes in the membrane potential is also very important because with sodium, potassium, and calcium ligand type, the membrane potential or the electrical characteristics of this are changed. Now, membrane deformation, such as mechanical pressure, sometime during this the membrane can be deformed. A mechanical pressure could exert and the membrane is closed. The second way is the selectivity by charge. That is, there can be a charge which has a different potential and positively lined pore allows anions through. So if in the channel there are positive pores, then the negative ions will pass through. Negatively lined pores allow cations to pass through. And this is how the cell regulates its ligand bound transformation, ligand bound the signaling of mechanism.